Your tattoo ink might not be as sterile as the label claims, and the FDA has the receipts to prove it. Tattooing is an ancient art form where we inscribe our bodies with meaningful images, using needles to breach the protective skin barrier and deposit ink permanently just below the epidermis. Ideally, that deposition process should be clean. You do not want to introduce possible pathogens into your bloodstream alongside your new artwork. For years, the industry has upped its game regarding sterilization, marketing products as sterile to reassure clients. However, a recent US Food and Drug Administration study found something surprisingly worrying. They tested a batch of 70 5-sealed tattoo and permanent makeup inks and discovered that a third of them harbored bacteria, some of which can make humans ill. That initial finding in 2024 was significant, but it left a critical question. Was it an anomaly or a widespread issue? To answer that, the FDA widened their net significantly. The agency conducted a comprehensive retrospective analysis of 4 separate FDA tattoo investigations conducted between 2018 and 2024. They looked at 259 unopened jars of tattoo and permanent makeup ink. The results were consistent and stark. A third of those 250, 9 samples contained bacteria. In total, they identified more than 250 bacterial isolates representing 94 distinct species. This confirms that the contamination found in the smaller study was not a fluke. It is a systemic issue. The data underscores that microbial contamination in tattoo and permanent makeup inks is widespread and diverse, reinforcing the need for sensitive molecular detection methods, expanded surveillance, and harmonized manufacturing standards to ensure product safety. According to a paper published in Scientific Reports, led by FDA microbiologist Ogyu Kwion, the most striking data point comes from comparing labeled versus unlabeled products. The researchers tested 118 tattoo ink samples that were explicitly labeled as sterile. Of those, nearly a third, specifically 30.5%, were contaminated with bacteria. For comparison, they tested samples without a sterility claim, and 35.5% of those were contaminated. The difference is relatively small. This means that even when a manufacturer claims their product is sterile, there is still a significant risk of bacterial presence. The label does not guarantee purity, and the industry standard for sterility appears to be more of a marketing term than a biological reality for a large portion of the market. Of the 94 bacteria species identified in the study, around a third, specifically 34%, are classified as potential pathogens. This includes organisms like Pseudomonas aeruginosa, Bacillus cereus, and Cutibacterium acnes, while the top comment section notes that these are common environmental bacteria. Their presence in an injectable substance is concerning. Pseudomonas aeruginosa is notorious for causing infections in compromised skin. Bacillus cereus is an opportunistic bacteria that can cause issues ranging from cellulitis to necrosis, if untreated. The presence of these specific pathogens in a product meant to be sterile highlights a genuine health risk for tattoo recipients, even if the majority of cases are mild. The scientific community and the public reacted with a mix of concern and skepticism. One commenter pointed out that the grouping of origin, United States versus everywhere else, is nearly meaningless. They argued that data by brand or manufacturer would reveal significantly more about what is happening. Another commenter noted that the study does not provide the source of the inks, leaving a massive flaw in the analysis. We cannot tell if every infected ink came from a handful of bad actors or if all brands have some issues. This is the only conclusive data that matters practically, and it is missing. Many hope this stir will lead to more rigorous third-party testing. The study raises the question of how inks become contaminated during manufacturing. The bacteria found are very common environmental bacteria. They are difficult to eliminate entirely from complex chemical mixtures. One commenter played devil's advocate, suggesting that it is safer for manufacturers to be vague about sterility to avoid lawsuits. This suggests that the term sterile is being used loosely. The data implies that the manufacturing process is not consistently sterile, even if the final product is packaged in sealed jars. The contamination likely occurs during the mixing of pigments or the filling of the containers, where environmental spores can survive and thrive. The takeaway is clear. Sterility in tattoo ink is not a guarantee, even on the label. The FDA findings underscore that microbial contamination is widespread. This reinforces the need for sensitive molecular detection methods and expanded surveillance for now. Consumers should be aware that the risk exists. The industry needs harmonized manufacturing standards to ensure product safety. Until then, the label is just a claim. The data shows us that a third of those claims might be wrong. It is a reminder that the barrier between art and infection is thinner than we think. The data is clear. Sterility is a claim, not a guarantee. Stay curious, stay safe, and we will see you in the next report.